Good morning. You're welcome to today's lesson. I want to hope that we are all okay. We've kept fine since last time, and you're continuing to keep safe. So here we are to continue with homeostasis. Remember last time, the time when we ended, we had seen how tissue fluid is formed, putting in mind that homeostasis is the maintenance of a constant internal environment, which internal environment we saw is made of blood and tissue fluid. But we said that if these are to be kept constant, there are organs which we call the homeostatic organs that are important in maintaining the constant internal environment. Among the homeostatic organs, we talked of the skin, we talked of the liver, we talked of the lungs, we talked of the kidney, and others. So today, we move forward and we begin by looking at some of these homeostatic organs, and we are starting with the liver. The liver is a part that we find within our body, and actually the liver is the largest organ in the body. It is really large, it is big within the body, and it performs a variety of metabolic reactions. Remember what metabolism is. So it performs a variety of metabolic reactions as the largest organ. At a point where it is found in the body, just like we see from that visual, it is at a point where it is, or it has a variety of what we call lobes. It has different lobes. The left lobe, the right lobes, the median lobe. There are about 5 lobes of the liver. However, within the liver, we also find it connected to what we call the gallbladder. It is connected to the gallbladder because you're going to see when you come to the functions of the liver that it forms what we call bile. But bile is stored within the gallbladder, meaning there is a connection between the gallbladder and the liver. So the gallbladder simply stores, and later bile is discharged through the bile duct to the duodenum in order to bring about digestive roles, just like we shall see later on when you come to the functions of the liver. Just next to it is the stomach and the esophagus and those other parts, just like we are observing at that particular point. But putting in mind that the liver is a major homeostatic organ and one of the largest, actually it is the largest metabolic center within the body. Now we begin by looking at the structure of this liver. You have dissected specimens, meaning you've seen the liver physically. Some of you have eaten liver from a cow. So we've seen what liver exactly is. But what is the structure of liver? What makes up this liver? Now liver, or the liver, is composed of numerous units which we call lobules. Each unit making up the liver is called a lobule. But we are noting that these lobules have structural roles. They even have functional roles, meaning they are important in making up the structure, but they are also important in enabling the liver to perform its functions. Structure of the liver. We look at it, we follow keenly and closely. We have seen, or we have said, that the liver is made up of several structural and functional units which we are calling lobules. Lobules are cylindrical. Now, When you look at the images that we're having, let's first look at the image on the left. The image on the left, we are having different lobules, different liver lobules. Do we see those cylindrical shaped structures? Now, let's concentrate on one cylinder, just like you're seeing. That one cylinder has a variety of structures, a variety of them. The cylinder is shown like a circle on top, but that top circle is divided into very many compartments, just like you can see. Each small compartment there, each small compartment is what we are calling a lobule. This means that a lobule, a liver lobule, is cylindrical, just like we're seeing there. Now, these liver lobules are many. They are many, they are cylindrical, and they are the ones that make up the liver. However, the liver is also made of cells. Remember, it's an organ, meaning it has tissues, it has cells. The liver is made of cells called hepatocytes. Liver cells are called hepatocytes. Now, hepatocytes being cells, it means that they comprise, or they have all the parts that make up a cell. Hepatocytes. Therefore, liver cells or hepatocytes, we are noting that they show no structure or functional differentiation. What do we mean? That they do not have particular specialized differences to make them perform particular functions or to have a particular structure. So they show no structure or no functional differentiation. Those liver cells which we are calling hepatocytes. But like we said, just like all other cells in the body, they have a prominent nucleus. They have those cell organelles. They have the Golgi apparatus. They have the mitochondria for production of energy. Remember, the liver is where most metabolism occurs. There is a lot of respiration that occurs there. There is provision of much energy or production of much energy within. Hence, the hepatocytes have numerous mitochondria. They have the lysosomes. They have the glycogen granules. Remember, sugars in animal cells are stored as glycogen. Therefore, the liver cells, the hepatocytes, numerous glycogen granules, fat droplets, and peroxisomes. If we remember the internal structure of an animal cell, these are the different organelles that we find within the liver cells. Now, peroxisomes in the liver, remember, they are responsible for production of catalase enzyme, which catalase enzyme is important in the decomposition or the breakdown of hydrogen peroxide. Hydrogen peroxide is one of the products of the many metabolic reactions that take place in the liver. But if hydrogen peroxide is left to accumulate within the liver, then it would end up being toxic, damaging the liver cells. That is why there are a variety of peroxisomes with catalase enzyme to break down that hydrogen peroxide. Now, in summary, going back to the liver structure, we are saying the liver is made up of several functional units called liver lobules that are cylindrical shaped. These lobules contain cells which we are calling the liver cells. Liver cells known as hepatocytes. Liver cells known as hepatocytes. Each hepatocyte, which is a cell, contains all the organelles that are found in animal cells. Namely, it has a prominent nucleus, the Golgi apparatus, the mitochondria, numerous of them, the lysosomes, mention them, talk about the granules, the fat droplets, peroxisomes, with too much catalase enzyme. However, the hepatocytes within the liver lobules are arranged in a chain. The hepatocytes are attached to each other in a chain, forming a chain of liver cells which we are calling the liver cord. The liver cord. Meaning, each cylindrical liver lobule is made up of several liver cords. That is, several chains of liver cells called hepatocytes. Now, when we look at our image on the right, the image on the right is a magnification of a liver lobule. It is showing us what makes up a liver lobule. Each lobule is made up of a chain of hepatocytes. That chain is what we call a liver cord. Therefore, each lobule is made up of several chains of liver cells or hepatocytes called liver cords. We've already seen the structure of hepatocytes. But in between the liver cords are numerous canaliculi and sinusoids. Canaliculi, plural. Canaliculus, singular. Canaliculi are bile ducts. They are small ducts where bile flows or is transported. While the sinusoids are small blood capillaries for movement of blood. There are for canaliculi for movement of bile, while sinusoids for movement of blood.